Okay, so this is the last section of the genetics, and we're going to talk about or switch from single gene disorders to chromosomal abnormalities. And you can see just from the slide here that leading cause of mental retardation and miscarriage is because of chromosome abnormalities. One of the tests that they've done is they found that 50% of the recovered first trimester spontaneous abortions actually have chromosomal aberrations in them. And I even put this little note in here that less than 1/3 of conceptions result in live birth, but I was just listening to XM a few weeks ago, or a month ago, and there was a doctor on there, on Doctor Radio, that was talking about how they're actually finding it's less than 1 in 5 of fertilizations will result in a live birth. So fertility and birth is a really tough thing, and a lot of the problems are that we have defects in our chromosomes, and those fertilized eggs, or now it's become a zygote or an embryo, will spontaneously abort because they don't have the proper genetics. So overall, we find that there are about 1 in 150 live births actually have some kind of chromosomal abnormality. So this is a quick refresher. A karyotype, what they do with a karyotype is they'll actually take the cell, they usually extract like a lymph cell, and then they'll catch it in metaphase of replication, and they kind of freeze it there. And then they'll shatter the cell, shatter the nucleus, and spread apart all these chromosomes. One of the things they can do is something called fluorescent in situ hybridization, which is FISH, is another shorthand, or the layperson term is actually chromosome painting, and do something like this, where they paint the chromosomes, and they can paint some of the genes on there too, and they can identify it. For the karyotype, they just line them up from the largest gene to the smallest gene, and then they put the sex chromosomes at the end. And that's why we name them. And one's the longest, second's the second, third is the second longest, and it goes all the way down until the sex chromosomes. You have to remember that when you're counting the chromosomes, a normal diploid cell has 46 chromosomes. 46. That's 2 pairs of 23. The first 22 of those are called autosomes, and the last one's called a sex chromosome. So altogether you have 44 in a normal diploid cell, 44 chromosomes that are autosomes, and 1 pair, so 2 chromosomes that are sex chromosomes. And those are called diploids. Diploids are your typical somatic cell, your body cell, but a gamete they're called haploid. You only have 1 set of those chromosomes, so just 23. Right? And then at the very end they have 1 sex chromosome. Remember, 22 autosomes, 1 sex chromosome, and that will help determine whether you're going to be a boy or a girl. Right? And of course it's that little Y chromosome that determines that. Alright, so the chromosomes, I just mentioned this or went through it. We just have to remember that the first 22 of 23 chromosomes are going to be autosomes. The last one's a sex chromosome. If you have a diploid cell, then of course you're going to have 2 sets of each of these. So 44 are autosomes and 2 are sex chromosomes, but they're paired up. Right. If you're an XX, you're a girl. If you're an XY, you're a boy. And we'll talk about what happens if you have malfunctions in the XX or XY department there. Okay, so some chromosome aberrations, some terms to get to know. First is euploid or euploidy, and I underline it for you. These cells have multiple of the normal number of chromosomes. So they have some kind of a multiple of 23. So it can be 23, it can be 46, it can be 69, it can be 92. All of those are multiples of 23, and they call those euploids. If it's 1 set, it's euploid, but it's haploid. That's in a gamete, a reproductive cell. Diploids, the typical somatic cell, are euploid, and they have 46. If you had more than normal, they call it polyploid, so many. So triploidy means 3. Tetraploidy, 4. And you can see corresponding numbers, right? If the fetus or the embryo is fertilized and it has more than diploid, so if it has 3 sets or 4 sets, it's not compatible with life and it will have a spontaneous abortion. And there are actually some cells in your body that have more than the normal 23 or 46. Like the liver will actually be polyploidy. It has some. Next, aneuploidy, of course, an being without. So this does not have a multiple of 23. It may have a multiple, maybe like 22 or 24, that would be aneuploidy. It may be 45 or 47, that's aneuploidy. So it's not the same as euploidy or polyploidy. Euploidy means you can divide it by 23. Aneuploidy means it's not divisible by 23. That's the easy, simple rule of thumb. So you may have 1 extra chromosome, not a whole set. So you may have that 1 extra chromosome, instead of having 46 chromosomes in your somatic cells, you might have 47. And that's an example of aneuploidy. All right. Aneuploidies, sometimes they're compatible with life, just having that 1 extra chromosome. If you have 1 too few chromosomes, it's not compatible with life. So the moral of the story here is it's better to have a little extra than a little less. And we'll talk about some things that have too many. in them. Right? So autosomal aneuploidy, partial trisomy, or chromosomal mosaics are some common ones. So partial trisomy means that you might have only a little extra part of a chromosome present in each cell. Trisomies are usually only common in like chromosome number 13, 18, and 21, and we'll talk about those three, or at least two of them. So having an extra chromosome 13 actually disrupts normal development, and it causes heart and kidney defects, so it's really tough for these kids to live. And one of the forms of cleft palate is because of a defect in, or a trisomy in number 30 or 13. Number 18, you can have trisomy in 18, but a lot of these actually died before birth. So very few of them live to be born, and if they do, they usually don't live beyond the first year of life. And it's because they have massive heart, brain, and spinal cord defects. Right? Chromosomal mosaics mean that only some of the cells in your body aren't the same. So you might have trisomy in some cells, but not all. And sometimes they refer to these people as chimeric. So the DNA in one of their cells is slightly different than the other DNA in the other cells. In this picture over here, you can actually see an example of trisomy. So here you have a normal karyotype, but if you look at number 21, there are actually 3 chromosomes, 21. And this is actually Down syndrome. Right? So let's talk about how this works. How you get too many copies of a chromosome actually happens when the chromosome's going through mitosis or meiosis. So if we're talking about replication and passing generation to generation, we're talking about meiosis, remember, individuals. And the process is called non-disjunction. Disjunction, during anaphase of the cell cycle, the chromosomes pull apart. So they just line up across the metaphase plate, and then now they're ready to rip, and then they rip apart, kind of like this. So you would see in this situation, you have the chromosomes together, and then they tear apart during anaphase. That's a disjunction. In non-disjunction, they don't tear apart completely. So here you can see non-disjunction right here, where they didn't pull apart and separate. So they didn't separate appropriately. So all of them went into one cell. This cell had no DNA, or no sets of that chromosome. So non-disjunction is where some of the chromosomes don't manage to pull apart appropriately, and they all stick in the same cell. Right. While we're talking about things like Down syndrome is the best example of trisomy. And it's trisomy number 21, like I showed you in that karyotype a few slides back. And this is one you really kind of want to get to know because it's a common birth defect. It results in about 1 in 800 live births. But what's interesting about it is that it's 1 in 8 live births, but they really predict that 3/4 of the fertilized eggs that have trisomy don't make it to birth. So about 3/4 spontaneously abort or are stillborn. So that's a lot of children that have trisomy 21. You know, even though only about 1 in 4 will actually live. All right, of those ones that live, about 20% will actually die before the age of 10. So of course we're talking about mortality right now. And then the rest of them that live beyond 10 will actually live to 60 or more years and have a pretty normal, quote-unquote, normal, healthy life. Right? Some of the signs or symptoms, one's low IQ. So average IQ is 100, their IQ can be 20 to 70, so lower than normal. They have a low nasal bridge, and they also have these things called epicanthal folds. And an epicanthal fold, the best way I remember it, is that if you look at like Asian heritage eyes, they have kind of an almond-shaped eye. The epicanthal fold is this area right here that gives them kind of that tight, like almond-shaped eye. Protruding tongues, so their tongue's a little bit thicker than their mouth area, and sometimes it wants to just hang out a little bit. They have short stature and poor muscle tone. So those are some common symptoms. And then one of the causes is actually an increased risk of maternal age. So this non-disjunction actually happens in most situations of Down syndrome. And I want to point this out now. Not all Down syndrome is caused by non-disjunction. About 97% is because of non-disjunction. So in other words, there's a 3% out there that's another cause, which we'll talk about in a few slides. So most examples are because of this non-disjunction. And that's trisomy 21. These symptoms I put down here are usually physical outward symptoms that you can see. There are a couple things that increase risk of like health problems. Like about 30 to 50% of Down syndrome children or people have actually congenital heart diseases. They also have a reduced risk for fighting respiratory infections, and they have an increased risk for leukemia. And we'll talk about that next week when we talk about cancers. So those are just some things you want to be familiar with Down syndrome. So I gave you some examples of prognosis. I gave you some outcomes. Even the etiology, in most cases, it's because of maternal age. And you can see The reason is because that egg in a mom has been sitting there for 35 or more years. It's been exposed to x-rays, toxins in the environment, things that are mutagens that can actually cause mutations, or things that are called clastogens, which we'll talk about, that cause chromosome breakages or chromosome changes. So those are just some examples with Down syndrome. Right. Another example of having too many is if you have too many of a sex chromosome. So one of the most common is actually trisomy X. So this little girl has 3 X chromosomes, and a lot of times they call it the meta-female. They just have way too much. Outwardly, appearance-wise, they usually look pretty normal, but some of the physiological things that can be problems, like when they're ready for menstruation, sometimes they don't menstruate properly. Sometimes they're sterile. Sometimes it leads to mental development problems, like delayed language development. Sometimes you can actually see the change in the brain on an EEG. Sometimes they have motor coordination problems. So they usually get accelerated growth until puberty, and then they slow down their growth. And they call this a meta-female. The more X's they have, the worse it is. And yet, it's actually possible for them to get 4 X's. So if you have this non-disjunction in mom, and the egg has 2 X's, and then you have the second set with non-disjunction, then she can actually get 3, and then dad donates 1, so you've got 4. So it's kind of an interesting issue. But most of the symptoms are actually like psychological symptoms. Like, I don't want to say attitude, but usually they have poor attitudes in school. They have like social issues where they don't get along with others very well, and of course that psychological bit with the intelligence kind of slows down. Some other issues that happen with sex chromosomes. So here's one. It's called Turner syndrome. So Turner syndrome is a form of aneuploidy. They have the 23 chromosomes. Or sorry, they have 23 chromosomes, but they don't have the double set. So they have 23 from one parent, and then they're missing 1 from the other. And in this situation, they're only going to have 1 X, and they call it Turner syndrome. Usually they're short in stature, so when you look at them, they have webbed neck, so a little bit more broad neck. They have what we call a shield chest, a very broad, very square chest. Now, typically we associate women with more of a curved shape going through here. Very broad nipples out to the edge, and a lot of times they'll get swelling and edema very easily. Right. Next one is Klinefelter syndrome. Klinefelter syndrome is 2 X's and a Y. Could actually be 3 X's and a Y, so it's potential. But the key here is that they have too many X's and they have a Y. So what sex are they? If they have XXX or XX and then a Y at the end, as long as they have a Y, they are male. Yep, so they're male. So they have male appearance. They develop female-like breasts. They actually have small testes for development. They're usually really tall, but they have feminine characteristics. So gynecomastia means they have breast-like tissue. They don't grow facial hair very easily. And they actually find this is pretty common. They just have that extra X, which gives them more of a feminine characteristic. And they're usually really tall. Right? So when they diagnose them, there are some signs when they're babies, like undescended testes might give a clue. A very small penis might give a clue. In children, if the child's having problems learning, they may be tested, and they have to do a karyotype to see this, and then they look. So they may be XXY, XXXY. But their symptoms are more feminine symptoms, or their problems, even mental development, increase with the more X's they have. And a lot of men out there don't know they have it. They're really tall guys. They don't grow facial hair very well. They may be a little bit less than 100 on their IQ scale. And they had no idea they had Klinefelter syndrome. So when they're an adolescent, you start seeing more feminine characteristics come out. Like they're not developing a beard like a normal male adolescent. They may start developing more breast-like tissue as an adolescent. So the key here is that adolescents, they should be supplementing it with a hormone. What hormone should you give them to make them more masculine? Testosterone. So these kids, they start developing more normally during puberty if you start giving them testosterone supplements. Right? And then as an adult man, if they want to reproduce, then fertility might be an issue too, because they have too much of the femininity going on. So taking testosterone supplements might help there too. Right? Some other problems, so abnormalities in chromosome structure, and here you can see a lot of breakages. So over here, you can see breakages in the chromosomes, weak spots in the chromosomes, and there are a lot of things out there that can actually damage. And here's a good example: clastogens from things like cigarette smoke. So when you're exposed to all kinds of toxins from the cigarette smoke, those things are clastogens and they weaken chromosomes. Right? So they can cause mutations. They can be mutanogens or mutagens, but they can also be class genes that weaken your chromosomes. And some other examples are like, I think it's pronounced acrididine, but anyway, acrididine is an antiseptic. It's there to destroy chromosomes, to destroy bacteria, but unfortunately if you get too much exposure to it, it can destroy your chromosomes. Benzene is a chemical that's in glues, paints, cleaners, cigarette smoke, car exhaust. When you breathe in too much of that, it's clastogen. So it can actually cause chromosomal damage. Ethylene oxide they use in hospitals as a sterilizer and a disinfectant. I mean, it's there because it's trying to kill viruses and bacteria by destroying chromosomes or DNA inside those bacteria and viruses. But unfortunately, if you get too much exposure to it, it can cause problems with you. So if you have breakages, here are a couple examples of what happens. You can have a breakage and actually have an entire gene deleted. So the deletion just removes the entire area. One of the diseases associated with this is called cri du chat, and it means cry of the cat in French. But cri du chat syndrome is when you break part of chromosome number 5 off. In the next slide, we'll show you some pictures. They have low birth weight, they have mental retardation, not mental retardation, and they have microencephaly. In fact, I put a couple more symptoms on the next page too. But here you can see an example of a deletion. So you break these homologous chromosomes here, one of them is broken and shorter than the other. You can have a problem with duplication, where you actually take part of a chromosome and you duplicate it. And the problem here is that when you turn one of that gene on, you're turning more on, so you're making more. Right? So duplication is usually less serious complications because you're just making more of something. And remember that whole it's better to have more than less kind of situation. Right? So you can have deletions and duplications, and we'll show you some more after we talk about cri du chat. But cri du chat, here you can see some of the symptoms. As this kid's developing, he has kind of a smaller head, so microencephaly. His IQ is a little bit lower. His growth rate is lower than usual. He has something called micrognathy, which means that the jaw is undersized compared to the rest of his, like his face. And sometimes they call it mandibular hypoplasia. But in this situation, you can see that the mouth doesn't want to line up right. And in most of the pictures, you see his mouth open, and you can actually see the teeth. What is really dangerous is actually this cardiac defects. So they can have heart issues. The cry of the cat just means that when they come out, they sound like a cat sound. And most of them will lose that by the time they're 2 years of age. But when you look at these symptoms, all of these kind of have similar symptoms or similar issues except cardiac defects. Most of these revolve around the head. So strabismus was talking about the eyes. So the strength of the eyes don't want to get them to line up and move appropriately, so they get things like double vision, the epicanthal folds we saw with Down syndrome, so development of the eye, growth retardation, things like that usually revolve around the head. And even when you hear the cry of the cat, it's because their larynx hasn't developed appropriately. So those are some of the symptoms. Right. Next, inversions. So you can break a chromosome like this over here, and what's cool about your chromosomes is that you have repair mechanisms that they see it's broken, they freak out, and they come back and they try and glue it together. Well, they're not that smart. They just see the break and they try and glue it. They try and fix it as fast as possible. So if this break flipped around and then it gets glued in place, everything's reversed. And the weird thing about this is, let's say you have a gene here that gets turned on. Normally, in a normal pathway, that gene's turned on, it's read along here, and it detects a new gene and it stops. Right? The problem is when you flip it over, if you read that same gene, you might read right through. Now, every time you turn this gene on, you turn that one on too. So you start making really weird products. And they call it the positioning effect because the neighboring genes are also expressed when you turn on one gene. So it's just kind of a strange thing. Right? And here you can see an example. So if this were the gene order where you'd have gene A, B, C, D, E, F, and G, you may have some flip in here where you break an area and now the genes read A, B, and then E, D, C, but F, G. So when you turn on this one, you might accidentally turn that one on in addition, or turn all three of them on. So that's just one of the common things that can happen. And the position effect is when you flip on an extra gene that you didn't originally want. Next is a translocation. Translocations are similar to inversions, where you break a piece of a chromosome, but in an inversion you put it right back in the same chromosome, just upside down. Translocation is when you take it out of one chromosome and you see these breaks in two chromosomes, and the DNA doesn't know where it goes, and like I said, the repair enzymes just come rushing in and they glue it back together. Well, if they glue it on the wrong chromosome, then you've got lots of issues. I really want you to take note of this one because Robertsonian translocation is the cause of the other 3% of Down syndromes. So 97% is caused by non-disjunction. the chromosome's not separating appropriately. But the last 3% is because of you have this Robertsonian translocation, where you'll take one piece of a gene, break it off, and flip it to another. In Down syndrome, what they find is that one gene is number 14, the other one's number 21. So 3% of all Down syndromes have this fusion of 14 and 21 where they switch parts and they fuse back together. Right? The other thing about those Down syndromes, if a parent has a child with Down syndrome because of Robertsonian translocation, they have an increased likelihood of having a second child with the Down syndromes too. So normal Down syndromes, like the 97%, it's a fluke. It just happens, you know. Older maternal age, there's some breakages in that egg that was released, and it's a fluke by that egg. But when you're looking at Robertsonian translocation, this is something that happens more frequently. Right? And then fragile sites. So fragile sites themselves, we talked about it a little bit before when you have a repeating sequence. So at the very end of a chromosome, you have this repeating sequence that gets longer. It makes it kind of frail and easy to break. And your book says no apparent relationship to disease, but we do know that there are some syndromes that are caused by this. So when we look at it, one of them is Fragile X syndrome. So it happens to the X chromosome, and you can see the normal chromosome length. You can see Fragile X chromosome, they have that weak spot at the very end. Right? So on the long arm of the X chromosome, you see this weakened spot, and it's because of a repeating sequence that goes CCG, 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 CCG. And of course, just like Huntington's, the longer that gets, the worse the symptoms are. And the symptoms are usually associated with like behavioral disorders, mood disorders, mental retardation. And the mental retardation, Fragile X is the second most common chromosomal cause of mental retardation next to Down syndrome. Right? All right, some of the treatments. So gene therapy, and this is just kind of like an overview. It's a really cool process. What they do is they take an adenovirus. And adenovirus is the virus that causes like the common cold, some respiratory infections, pink eye, bladder infections, things like that. It's a pretty common virus. But they take that virus, and what happens is they'll put the good DNA in it, and then they give it to you. It's a virus. It tries to get into your body and merge itself with your DNA. The problem is that it's a DNA virus, and we talked about this before a little bit. A DNA virus that gets into the cytoplasm is more easily attacked by the cell, so it's harder to get it into all your cells. The second big problem is that when you give somebody this adenovirus, what's their immune system going to do? It's going to attack it. It's going to know it, and the more times you give them this virus, the faster it's going to attack it, and the more responsive it's going to get. So repeated inoculations are important to help try and get into as many cells as possible. But the problem is that they become immune to it. Right? So they use other transport mechanisms like liposome delivery, so they put it in liposomes. They can do it by, I'm having a brain fart on the term. I'll get back to that. They can do it by transferring genes, basically. So they can give you something that will inhibit a bad gene or turn on a good gene. I can't remember the actual term for that. There's a name for it. And then the ideal time to do is immediately after fertilization, because right after fertilization, there are only a few cells. So if they can fix those cells right away before they develop into more and more cells in the whole organism, then they have a greater likelihood of actually being able to treat it or cure it. So another problem that happens right here, environmental factors. Things like diabetes during pregnancy. And you already know that diabetes can cause things like abnormal weight in babies, giving big fat babies. Smoking can cause it. Smoking causes low birth weights. Okay. Fetal alcohol syndrome. This is actually a picture of a thalidomide baby. So there was a chemical called thalidomide back in the, I think it was the 50s or 60s. They gave to people so that they wouldn't have morning sickness. Well, it was good for the mom, but unfortunately what happened is that it caused what they refer to as flipper limbs on the baby. So either they didn't have full limbs or they would have these limbs that had malformed, like fingers and hands on them. And anything that causes this to change in birth or change like this in birth, they're referred to as teratogens. Teratogens change what's going on in the embryonic development. So the most important days where teratogens can be exposed are actually day 15 through 60. And I'll show you. So here you can see days 15 through 60, you're developing most of the major organs. So here you can see the central nervous system, here the heart, here the extremities, the eyes, the genitalia. Most of those are happening in the first couple, or they're developing in the beginning in the first couple weeks to first month or two. And then, oops, I skipped over one. There we go, the last homework question. So the last homework question is epigenetics. And epigenetics is kind of unique because epigenetics, you don't change the DNA sequence. The DNA sequence is still exactly the same. What happens is you turn off or turn on the genes differently. So I'm going to have you look it up. Epigenetics, and the perfect example, I think, is these two, Angelman and Prader-Willi syndrome. These two disorders are both caused by exactly the same gene, but epigenetics will turn on or turn off the gene in a different way. So what I'm going to have you do is I'm going to have you look up these two syndromes and tell me what's the name of the gene, what are the symptoms of Angelman, and how does epigenetics cause this? How do they believe epigenetics cause it? Then you look up Prader-Willi, what's the gene, and I already gave it away, it's going to be the same gene. What are the symptoms, though, of Prader-Willi? How do they think epigenetics causes Prader-Willi? And you might see some links, and I'm kind of hinting around that where the genes come from, mom or dad, might influence it too. Even though it's the same gene, depending on the parents, epigenetics may flip it on or off. So these epigenetics are interesting because they can look like heritable disorders, like something that is passed down family generation to generation. But they can also change the way things work later in life, like cancers. It can affect your immunity. It can affect things like schizophrenia later in life, mood disorders, depression. So next week we're going to start talking about cancers, and we'll come back and talk a little bit more about epigenetics. But for now, I just want you to look these two up. So you can pause it, and there's actually one slide. You don't have to do this slide. I'm telling you right now, you don't have to do this slide. But if you're interested in epigenetics, there are a couple of resources that are really cool, especially this last documentary, The Ghost in Your Genes, because it's a really cool explanation of how your environment actually affects your genes. Whether you have good, healthy genes or flawed genes, your environment can actually affect it. So that's all for this week, and I'll see you next week.